Hello, this is experiment number 7 called Culture Transfer Techniques. The purpose of this experiment is to learn how to subculture microorganisms in different types of media. Let's first understand what is subculturing. That means transferring the microorganisms from one media to another. And what is a media? Are different places where we set the microorganisms to grow, such as displays. We have the natural mediums like the forests or the oceans, but we are limited. We don't have those in the labs, so we have to use different places that are these or these, so our microorganisms can grow or react to experiments. What media do we have in the lab? In the lab, we are going to be working with 4 different types of media. The first one is called agar slant. This is what it is. It is a test tube, and we see that what we have inside is a nutrient that is solid and is placed in a way that has a surface that is slant, and that is why it's called agar slant. We want to have another kind of media that is called agar deep, and we'll see is also a tube, a test tube, and we have also a solid content that doesn't move, but in this way doesn't have an slant, it's just a straight surface. Then we have the broth tubes, and again these are test tubes, and the media that we have is a liquid, you see, and here we can put upside down because it's going to drop. The last one that we are going to be working with is called agar plate. See? It's an agar plate, and in this case we already have some bacteria, and it has a solid media inside where it grows. Now let me introduce you with our inoculating tools. First one is the needle, second one is the loop. You can see the difference. This one is thin, and this one it has a larger surface. And when we work, there are 2 principles here. One is the tension of the surface that we're going to be working with, and we don't want to pull too many or too little bacteria. We want just to have the correct amount, and based on where we start and where do we finish, we're going to be selecting one tool or the other. And the second one is going to be sterilizing. It's a very important thing we have to do before and after, and it's basically resetting our tools. To sterilize, we're going to use our incinerator. We're going to put our inoculating tool there, and we're going to wait for a few seconds, like 10, 15. It might become red because it's hot. And after that, we take it out and we let it cool down for a few seconds before working with our bacteria. Let's talk now about the goals for this experiment. The first one, we're going to practice how to inoculate. We're going to be using our tools, and we're going to practice the different steps. The second one is going to be to subculture the different bacteria in different media, and we're going to see how the bacteria grows and develops in different media. And then we're going to incubate, give some time in a good temperature to these bacteria. And finally, we're going to observe at the end of the experiment how the bacteria grew, and we're going to complete a chart, and we're going to practice our analytical skills with this. Now, let's talk about the material we're going to use for this experiment. The first one is our incinerator. And we're going to be using our needle and loop, the inoculating tools, and we're going to be using the nutrient agar slant, the nutrient agar dip. We're going to be using a broth tube, and we're going to be using the Serratia marcescens, that is going to be the culture that we're going to be working with. Let's work now on our procedures. The first one, we're going to be working in 3 different steps. And in the first one, we're going to transfer from a broth to a dip using a needle. In the second one, we're going to transfer from broth to slant using a loop. In the third one, we're going to start with our agar plate, and we're going to transfer to a broth using a loop. Let's work on our procedure number 1, and in here we're going to, first thing is going to be to label our media, and we're going to start with the broth and we're going to finish in the dip. So what we're going to do is to label our dip. I'm going to place a tag, and I'm going to put the name of our culture, that is going to be Serratia marcescens. I will put an S, an M, and I'm going to put from broth to dip, just to have an idea of what we are going to be working on. And then when we see the results, we don't get lost. Okay? So the first thing what we're going to do is we're going to start with our broth. Our broth is liquid. It contains the Serratia marcescens, and our end is going to be solid. Okay? So an important thing for inoculating is just using the Goldilocks principle, not too much and not too little. So here we're going to use our needle. Why don't we use the loop here? Because if we insert the loop in our solid, it's going to be too much and it's going to be trapped. So what we want to do is just to use our needle. So we're going to start inoculating the needle. We're going to wait for a few seconds until it warms up. Give some time. Now we're going to be waiting for like 20 seconds. And what we are going to do is get our broth containing our culture, and to prepare both tubes, and I'm going to insert the needle, and I'm just going to place it inside, ensuring that I'm getting the bacteria from this media. When I get it, what I'm going to do is transfer it to our solid media. So what I'm going to do is stab our media. Then there it is. Now you can see I'm going to place the cap, the media, not too tight. So it's going to be loose to let some oxygen come. Now what we're going to do is to inoculate our needle, to sterilize, the needle. Our second procedure is going to be to transfer the bacteria from the broth to the slant. And in here, we're going to have a big surface. So we're going to use our loop. So we grab the bacteria from the broth, and then we're going to strike it over the surface to spray it. Let's give some seconds for our loop to warm up and to kill any bacteria, to keep our colonies pure. Now I'm going to wait for 20 seconds. Meanwhile, I'm going to get our two media prepared. So now I'm going to insert our loop in our broth liquid media. Just going to move it so I grab the bacteria. One is here. I'm going to place it in our nutrient agar tube by striking it over the surface. See? There it is. See the marks at the left with the loop. Now I'm going to sterilize our loop, and I'm going to place the cap over the media, again loose, to let some oxygen exchange. Now we are going to do our third experiment. In this one, we're going to transfer the bacteria from the agar plate to the broth. Okay, so in this one, we're going to use the loop as well. I'm going to get our bacteria. I'm going to get some with the loop. See? Here. And now I'm going to get our broth media, and I'm going to place the loop inside and spread the bacteria. Some is hanging in the loop, so I have to shake, ensuring that some bacteria are moving to our media. I still have some in the loop, so I keep shaking. Now I have most in our liquid media. See? And here the results of our work today, and see how we are labeling everything. And here we have the name, the number of our table, and our tubes with the labels, SM of Sriracha marcescens, and then I put initials just signaling the media that we started with and the media that we finished. Here we're back to analyze the results of our experiment. We have here the tubes. The first one we're going to check is our nutrient agar deep. Here it is. You can see the results. The growth. We have red bacteria, the Sriracha marcescens, and you can see how it grew on the top and where the needle make a hole, leaving some oxygen and air to come. And we're going to proceed and sketch this into the chart. The second one is the nutrient agar slant. Here is the growth. You can see it grew over the surface. That tells us one thing, is an aerobic bacteria. And finally, we're going to see what happened in the broth. And you can see it's turbid and it's more on the surface. We have some in the back, but is not the growth, are probably dead bacteria. The growth is more on the top.